As you might guess from my background, I haven't actually been to the Ear Institute since March, which is kind of crazy because my lab is based in a different place. So it's about a mile away. So when I have been into London, I haven't actually been to the Ear Institute. So it sort of feels slightly weird to be associated with it almost at the moment. Anyway, I am really pleased to be here talking to you today about some of our work looking at how auditory cortex represents timbre. And normally I say timbre, but I feel with this audience, I really need to qualify that I mean spectral timbre. And I probably don't need half of these introductory slides either, because I'm sure you all know that spectral timbre is a key cue for identifying a sound source. So whether you're listening to human speech, so for example, a vowel sound where it's the spectral envelope that allows you to distinguish, for example, an ah from an ear, or whether you're listening to music and distinguishing different musical instruments based on their spectral envelope, of course, also on the temporal aspects of timbre. That's also critical for musical instrument identification. But today, really, I'm kind of reducing this down to spectral timbre and the spectral envelope, mostly of vowel sounds. So I'm sure you know, but the ear detects sound waves come in, they hit your eardrum. Those vibrations are amplified by the malleus, the incus, and stapes. And those vibrations then travel to the cochlea, which is responsible for translating the acoustic energy in a sound wave into neural activity that your brain can make sense of. And a key feature of the cochlea is the fact that it's mechanically tuned. So essentially, an incoming sound wave is decomposed down into its separate frequency components, such that individual auditory nerve fibers signal particular sound frequencies. So the representation that the brain has available to it of an incoming sound mixture is essentially its frequency content. And that frequency representation is continued all the way up the auditory pathway. So this is a human auditory pathway here. So we have the cochlea, a series of subcortical processing stations extract specific cues about a sound, and eventually the sound ends up in the auditory cortex, which sits kind of on your temporal lobe, not so far from kind of behind your ear. And even here within the auditory cortex, neurons can be defined according to the specific range of frequencies that they respond to, and they sit essentially within a map, such that within a cortical field, you'll have neurons that respond to high frequencies at one end of that map, and neurons that prefer low sound frequencies at the other end of a map. So why is auditory cortex an interesting place to study? Well, once you get up here, neurons start to integrate information across a broader range of sound frequencies. And if you're interested in something like the spectral envelope of a sound, it seems critical that you're considering neural activity across a broader range of frequencies than you might find in, say, the kind of cochlear nucleus. So we think auditory cortex is an interesting place to look at. It's also quite convenient because it's on the surface of the cortex. We can find it easily. So the second question, so this is a human auditory cortex. And of course, we are humans. We are interested in human sound perception. But there are, of course, experiments that you cannot perform in a human. There are techniques that are not available to you unless you use an animal model. But I'd also like to argue that actually studying animal models is interesting in and of itself. Almost all animals vocalize. So whether you're vocalizing to search for a mate, to communicate within your group of conspecifics, to communicate with your children or your parents, vocalizations are really key. They're key to almost all species survival. And many vocalizations have some sort of rich spectral envelope within them. So it's not absurd to kind of consider the idea that spectral timbre might be a perceptual attribute that's accessible not just to humans, but really much more broadly across the animal kingdom. So I'm going to talk today a little bit about what we've learned about how the ferret, which is the animal model we use, actually processes sound timbre, and try and convince you that ferrets at least are quite a good model for how humans process sound timbre, and that a lot of the mechanisms that we use are probably quite strongly evolutionarily conserved. Okay, so the outline for today, sort of a talk in three parts. Firstly, I'm going to talk about some older work which took place in Andy King's lab at the University of Oxford, working with Kerry Walker, who was a postdoc there with myself at the time, looking at how the brain might support perceptual constancy generally, but specifically in this case, how you might be able to extract invariant representations of sound timbre in the face of changing other parameters, such as its pitch or its location in space. In the middle of the talk, we're going to look a little more deeply at the cues that our animal model used to perceive sound timbre and compare that to humans in a very similar task. And in the final bit of the talk, I'll present some more recent work where we've actually been exploring perceptual constancy and its neural correlates in animals as they perform these kind of acoustic discriminations. So kind of big picture theme of the research within my lab is really how are acoustic parameters translated into perceptual qualities. So if we think about sound, there's kind of three key perceptual attributes that people might use to describe a sound source that you're listening to. So the first one of these is its pitch, which we know is underlined acoustically by its periodicity. The second, and the one that's interesting to us today, is the spectral envelope, which gives rise to the spectral timbre of a sound. And the third acoustic cue might be binaural cues that give you a perception of where in space a sound arises from. So the questions we set out to answer in this study was, are there neurons within auditory cortex that actually represent any of these perceptual attributes independently of others? And sort of secondly, if we zoomed out a little bit, could we see that there was particular specialization within a given cortical field? So by timbre selectivity, so we're sort of going to rephrase this question for the purpose of today's talk, which is, is there timbre selectivity within auditory cortex? And by selectivity, what I really mean is, are there neurons that can tell me what the timbre of sound is, even when I change the pitch or the location in space? Do we have a representation, a neural representation, that's effectively invariant to those changes in those other parameters? And the approach that we took was to parametrically manipulate the sounds across multiple perceptual dimensions. Typically in neurophysiology, what we tend to do is to map the sensitivity of a neuron to changes in one stimulus dimension, and that tells you something about its sensitivity to that dimension, but it doesn't tell you how selective it is, because it may be that actually if you change the sound along some other dimension, you get a much larger change in the neural response. So here we had a stimulus set that comprised of 64 artificial vowels, which was essentially all possible combinations of 4 pitches, 4 vowel identities, and 4 spatial locations. We chose these artificial vowel sounds, shown here against a natural spoken vowel, for a variety of reasons, but mostly because we could perform these kind of parametric changes within them. So we could determine the periodicity of the vowel, determining in turn its pitch. We could determine the timbre by changing the position of these formant peaks that give you effectively the spectral envelope that the listener will use to determine the identity of this vowel. And by presenting them in virtual acoustic space, we could move the vowels around the animal's head. So our prediction is that timbre selectivity would be observed as a neural response that was tuned to a particular vowel or to particular vowels, and whose response was uninfluenced by changes in pitch or spatial location. So, okay, this is a human brain. If we zoom into this human brain, or indeed the ferret brains that we were actually recording from, you'll see that they're comprised of millions of neurons, individual nerve cells. Things we're interested in are these very rapid deflections here that we call action potentials or spikes. And essentially, this raw data is changed into a series of times at which these spikes occurred. And that gives us data that looks something like this. Okay, so I'm going to walk you through this slowly. So we have 64 stimuli. So we have 64 different boxes here. And within each box are the responses of the neurons, so the times at which the neuron spiked, depicted as a dot for 40 presentations of that one sound. The sound was 150 milliseconds long, so you can see this sort of onset response that occurs, and an offset response in some cases. And here I've taken the 64 stimuli and I've organized them according to where they came from in space. And this is a reasonably typical auditory cortical neuron, because sounds that are coming from the opposite side of space to which we recorded, so from the right-hand side of space for a neuron that was recorded in the left brain, elicit, in general, stronger spiking responses than sounds that come from the same side of space as the neuron. We can take exactly these same data, so again, these responses to these 64 stimuli, simply reorganize them. So now I've shuffled them around so that they're grouped according to their pitch values. So you can see high pitch responses to high pitch sounds at the top, responses to low pitch sounds at the bottom. And you can see again that this neuron seems to prefer high pitch sounds to low pitch sounds, but it's not responding uniformly throughout this, this block of sounds that have identical pitch values. And we can kind of complete the circle by again, shuffling them so that they're now organized according to their spectral timbre. And again, you can see that this neuron seems to really not be responsive to this particular vowel very much at all, and respond more strongly to the a and the u than the e. So, but even again within these preferred vowels, you can see that there's this shift from responding slightly more for locations to the right of the animal, and less to the locations on the left. And again, within each block, you can sort of see that there's also an influence of pitch. So this is just one neuron. It's an example, but it's a very typical one of the sort that we recorded, in that it's modulated quite strongly by all three stimulus dimensions. So this neuron definitely does not pass our tests of being timbre selective. It's sensitive to timbre, but also to pitch and to location in space. And actually, the vast majority of neurons that we looked at fell into this category. So nearly 40% of neurons were represented by at least two of the stimulus parameters. Just over 30% were actually modulated by all three. So we actually found relatively little evidence of specialized populations of neurons that were actually selective for single attributes. When we stepped back and asked where do we find neurons of each response type across the surface of the cortex, we also didn't see much evidence that particular cortical areas were specialized for any of these three attributes. So this is a schematic of a ferret brain. So sort of zoom into auditory cortex here. Auditory cortex is color-coded according to its frequency selectivity. So high frequency areas are shown in red at the top here. Low frequency areas are shown in blue. And these reversals, where you have areas that go from kind of red to blue to red, are what we use to indicate the boundaries between cortical fields. Shown below are kind of tessellation maps of all of the neurons that we recorded, color-coded according to how sensitive they were to each of these three sound dimensions. You see, like, we can zoom in on, for example, this neuron here. It's very sensitive to sound timbre, not so much to pitch, but it's also sensitive to changes in spatial location. But in the vicinity of this neuron, likewise, are neurons that are sensitive to pitch and are not sensitive to timbre, for example. We have all possible combinations. So we really took this as evidence that there was a really distributed representation of these perceptual dimensions throughout auditory cortex. You could argue maybe this is a weird sort of ferret thing. So we were really reassured when a human functional imaging study from Andy Oxenham's lab was published a couple of years ago, looking at the distribution of pitch and timbre sensitivity using relatively similar approach to ours, and found really very similar, like strikingly similar findings in terms of sort of distributed interdigitated selectivity or sensitivity to pitch and timbre in the human auditory cortex. Okay, so single neurons are coding multiple perceptual features, but is it possible that they're doing so at different times? Because if the neural sensitivity is mixed, then how are you as a listener able to disambiguate a change in timbre from a change in pitch, for example? So one possibility is that this is a sort of emergent property of a population code that we don't see when we look at the single neurons in this manner. Another possibility, which isn't mutually exclusive particularly, might be that the encoding of particular perceptual dimensions are unambiguous, but only at particular points in time. So this was a hypothesis we, or specifically Kerry, set out to address with this data set. So typically people take spike responses or calculate information metrics across a reasonably long time window that spans the duration of the sound. And what Kerry wanted to do was to actually divide this up now and instead compute the mutual information, which is essentially just a measure of how certain we can be about what stimulus was presented based on the spiking response. Now for discrete time windows. So we have one time window that was at the onset of the sound, one that was sort of targeted the sustained response, one at the sound's offset, and a late window as well. And we measured the amount of information that was available in each of these time windows and plotted them for each of the three sound dimensions at each point in time for each dimension across each of the 5 cortical areas that we were interested in. And what you can see here at the population level is that each perceptual attribute has its own distinct time course in which information is available and kind of passes through these different auditory cortical fields. So timbral information occurs very early on in the response. It's primarily in the onset of the response, and it's most strongly represented in the primary fields. Pitch is also present in the onset, although if you note, there's actually really, there's quite different scales here. So the information about pitch is quite significantly less than about timbre at the onset, and it's most strongly represented in the sustained response in secondary cortical fields. And location shows it is also quite onset dominated. So this is a kind of global picture of what's going on. We can see this even in the responses of single neurons. So these are just 4 different single neurons recorded with the amount of information that's available about either the timbre of the sound or its pitch plotted across time. And you can see that in each of these examples, the timbre information is occurring earlier than pitch information. And that actually, if you know when to read out activity in each of these neurons, you can have a pretty unambiguous idea of whether the change in spike rate that you're observing is due to a change in the pitch of the sound or a change in its timbre. And this sort of just shows you an average response for primary cortical neurons, again, emphasizing this, this idea that timbral information is occurring earlier. There's a sort of interesting behavioral follow-up to this that I don't really have time to talk about today, but the ferrets, if they're trained in a go, no go task, can actually perform this discrimination faster if it's based on the timbre of a sound than if it's based on the pitch of a sound. So it really seems like this is this neural correlate, or this is a neural correlate of something that's behaviorally meaningful. Okay, so sort of summary part 1. We've seen that neurons are sensitive to multiple perceptual dimensions, and that this information appears to be distributed across cortical fields. And in turn, we've also seen that temporal multiplexing potentially allows you to disambiguate changes in different, within the same neuron in response to different perceptual dimensions. But this leaves some questions unanswered. So firstly, do we not see neural correlates of perceptual constancy simply because ferrets don't experience perceptual constancy? This would be a reasonably kind of boring explanation to our data, but one that at this point we couldn't rule out. And secondly, all of the recordings that I've presented to you so far were made in the auditory cortex of anesthetized ferrets. It seems possible that neural correlates of perceptual constancy might require engagement in some form of task, or at least being awake and alert and listening to the sounds. So we're going to do 2 things now, which is switch from our kind of sleepy anesthetized ferrets to an awake behaving animal. But first we're going to do some psychophysics and try and understand to what extent the ferrets are capable of experiencing invariant representations of timbre. So I believe that ferrets are really excellent animals for timbre perception. And that's because firstly, they share a similar hearing range to humans, and in particular, they have really good sensitivity to low sound frequencies. That's really important if you're interested in things like how humans use pitch to segregate two competing sounds, or use ITDs to localize a sound. Ferrets are very vocal animals. They're quite happy and chatty. And one of the vocalizations that they make, which is called a duke, actually shares quite similar properties to human vowel sounds. So you can see they're composed of these very short syllables, but each syllable has a pitch to it, and it has what look like formants in it. And possibly because of these first two things, we found that ferrets really readily learn timbre discrimination tasks much more quickly than, for example, a pitch discrimination task, or more complex tasks based on other, perhaps more abstract features. So the task that we trained them in was this. So it's a 2 alternative forced choice task. So the animal pokes its nose in the center go spout. That triggers the presentation of 2 sound bursts of an identical vowel. Again, these artificial vowels that we used in the recordings. And in this case, the vowel could either be an oo or an air, and the animal was trained that if it heard an oo, it should go to the left-hand spout, in which case it was rewarded. And if it heard an air, it should go to the right-hand spout. So ferrets were able to learn this task. We trained them with vowels that have a single pitch, and we then tested them across a variety of other fundamental frequencies. And the animals were really able to generalize very well across a whole range of different pitch values with really minimal additional training. This, again, was work that we carried on, that we started in Oxford, but that Stephen Town, a postdoc in my lab, really picked up the reins of when we set up our lab in London, and really started to get to the bottom of how the ferrets were using particular cues to make their timbre judgments. So, and to do this, we created a stimulus set that essentially put different acoustic cues in conflict with one another, so that we could see which of the cues the animals followed. So animals were trained either to discriminate oo from air or ah from ear. So each vowel has a formant, a first and second formant peak associated with it. You can also determine its spectral centroid. And you can also measure the distance between the 2 formants, so the interformant distance. And it's possible that the ferrets are picking up on any or all of these cues when they learn this discrimination. So to tease apart what cues they might be using, Stephen created these mismatch sounds. So the first kind of set of mismatch sounds simply take the first formant of one vowel and put it with the second formant of the other, and make the reverse contingency as well. So the F1 of the air paired with the F2 of the oo. A second set of sounds took the interformant distance from each vowel and anchored it to the first formant frequency of the opposite vowel. And a final set took the interformant distance and anchored it to the second vowel, although that yields some mismatch vowels that are impossible because they would have a negative first formant frequency, so we don't have complete sets for them. We can also compute for each of these mismatch vowels what their spectral centroid is, and it's quite tightly correlated with the second formant frequency. So, oh, that's a shame. This was meant to be animated. Pretend you can only see the top bar. Okay, so these are data from a single ferret for all of these mismatch vowels. So in the top are the trained vowels. So the animal's trained to go left for an oo, right for an air. So you can see that when we present the oo, the animal goes left almost all of the time. When we present the air, the animal goes right almost all of the time. We can conclude the animal knows what its job is. So far, so good. So in the first set of mismatch vowels, we swap the first and second formant frequencies, so that in order to ask which, when we put these 2 cues in conflict, what does the animal do? And you can see quite clearly here that the animal follows the position of the second formant. So when the second formant matches the air, the animal goes right. When the second formant matches the oo, the animal goes left. These next sets are the vowels where we measure the interformant distance to the oo and anchor it to the air's first formant frequency, and vice versa here, and the same but anchored to the second formant frequency. I don't know where that text has come from. That's weird. There's something, there's a ghost in the machine with this slide. So what I hope you can see is that with these interformant distance simile, the animal's behavior is much more ambiguous. It's quite tricky to see. I mean, basically the animal's guessing in both cases. In the case where we have the second formant from the air with the interformant distance from the oo, you can see that the animal goes quite strongly right, indicating that it believes it to be more air-like than oo-like. So in order to make sense of these data, what we did was to measure D prime values, asking if we have the behavioral responses from the animals, how well can we distinguish the sounds that they heard? So in this case, we have, if, for example, we were going to compute the D prime between this vowel here, the trained air, and the mismatch vowel that has the second formant of the air, the animal goes right nearly all the time in both cases, so we'd yield a very low D prime value for this because we can't distinguish these two sounds based on the response. In contrast, if we compared this mismatch vowel to the trained oo, we get a very high D prime because the behavioral response is very clearly different. So when we so here essentially what we're doing is plotting the D prime according to each of the acoustic measures that we have. And what I hope you'll agree is that really it's the change in second formant frequency that seems to be driving their behavior. I'm wondering if someone's accidentally annotated the screen, because I don't know where this blue seconda formante has appeared from.
Sorry, Jenny, I think that's probably what it is. So please do not.
It has vanished.
Thank you very much.
Thank you. So in order to kind of make sense of this a little more clearly, Stephen performed a series of logistic regressions. And essentially what he started with was with single parameter models in which one acoustic cue was included in the model. The way that you interpret these data is, so these are deviance measures. So a constant model has a certain amount of deviance associated with it. When you add variables to your model, you should see a drop in the deviance. That indicates it's a better model. So the F1 frequency is poor at predicting the animal's behavior. The F2 predicts it quite well. The interformant distance, which correlates with the F2, slightly less well. Sorry, the interformant distance does not correlate with the F2, but it explains it a little bit better than the F1. Spectral centroid, which correlates with the F2, also does a reasonable job of explaining the animal's behavior. We can create increasingly more complex models, but in this case, they don't actually buy us any more explanatory power. So a model that includes just the second formant frequency really does a good job of explaining the ferret's behavior. And this, for people who work in human timbre perception, may be raising red flags, because we know that when humans are making judgments based on vowels, we certainly use more information than simply the second formant frequency. So to kind of get a better handle on this, we tested humans in as close a way to the way we test animals as possible. So we gave them a laptop rather than a booth. We gave them smileys on a screen instead of free water. But essentially, we didn't give them any instructions, and they simply had to, by trial and error, work out that they had to press one button when an oo was presented and another button when an air was presented. And then we gave them these mismatch responses and modeled their behavior in the same way. And actually, we had really quite a similar pattern of responses. The individual human listeners themselves were more variable than our ferrets, but the second formant frequency did a much better job of explaining their behavior than the first. And the pattern across these single parameter models was very similar. And again, there wasn't much to be gained by including ever increasing additional parameters in the model. Okay, so interim summary number 2, we think ferrets, and I hope by now you think ferrets, might be a good model for spectral timbre discrimination, and that in the context of this task, which is obviously much more limited than the way that you and I might process vowel sounds in the real world, their behavior is highly consistent with human listeners. And when we trade different cues off against each other is the second formant frequency that's particularly important. So we pursued this task, but now we're going to go back to recording auditory cortical activity in the context now of animals that are actively discriminating these sounds. I'm afraid I haven't got a video for this because I couldn't get the sound to work, which is a problem because the main videos are actually on an old computer in the Ear Institute, which I can't access at the moment. Such is the joy of the times we're living in. So in this experiment, as previously, we had, we trained the animals at a fixed pitch to discriminate pairs of vowels. So here again, oo and air. We then tested them across a variety of different pitches from different spatial locations at different sound levels. And finally, with both voiced and whispered vowels. So the whispered vowels we generated by taking the spectral envelope that we applied to the harmonic sound that we started off with to create the voice stimuli, and instead passed a noise burst through it, so that we ended up with sounds that at least the humans are readily discriminable as sounds with a vowel-like identity, but that lack the harmonic structure. And when we tested the animals in these sounds, we saw that they could indeed discriminate across a range of pitches. Location was irrelevant at all, but the very quietest levels, which given the sort of level of background noise we sometimes have in the lab with the number of computers and stuff we have running is potentially not surprising. Performance was flat, but we did see this dip with the voiceless vowels. So in the case of the whispered vowels, 3 out of the 4 animals could perform the task, but they did so at a much reduced level of performance. It's worth saying that these voiceless vowels were presented as probe trials. So the animals heard them very, very rarely. And when they did hear them, they were randomly rewarded. So there's a sort of question here about to what this, this is a snapshot of their natural behavior. It's not to say that they couldn't do this task if we pushed them harder on it. What I haven't shown you here is some extra analysis that Stephen did, which looked across the cohort of animals we have now who've done this task, which is quite a large number, and showed that actually their performance on their first experience of most of these other parameters was reasonably indistinguishable from their performance that you see plotted here. So the animals do seem to generalize across these different stimulus dimensions very naturally with really minimal additional training. So does neural invariance emerge doubly perhaps when animals are engaged in a task that actually requires perceptual constancy? So we have here a task where we've really got 3 nice examples of perceptual constancy, and another example that's actually quite useful to us of a case where the animals are not showing perceptual constancy. So again, I'll start with a single neuron example. I'm going to sort of give you the relatively quick summaries from this study. If you have kind of detailed questions, do feel free to get in touch with me afterwards. So this is a single neuron, the same neuron in each case here. And you can see at the top the rasters. So these are, again, the dots are times the neuron fired. Each row is a trial. And the trials are grouped according to the identity of the vowel. So the green trials are airs and the black trials are oos. And across this whole set of sounds, these sounds are varying in pitch. These sounds are varying in level, in location. These are varying in level. And these last ones are varying in voicing. And you can see that if you zoom in on the onset response here, in each case, the green sound, the air, is eliciting more spikes at the onset than the oo. So this looks like a good candidate neuron for some kind of invariant representation of timbre. We can also, again, sort of play the same game we did earlier, take exactly the same responses and reorganize them according to now the nuisance parameter, if you like. So here, sounds of different pitch, different locations, different levels, and whispered versus voiced. So despite the fact we have information here that looks like it is encoding the identity of the vowel quite nicely across these other changes, at other time points in the response, again, we see this joint sensitivity occurring. So in this study, what we did was to perform a decoding analysis that asked, firstly, is information present at all? And if so, like what's the magnitude of the information? But also, Stephen developed a sort of optimization method here with the decoder that allowed us to say not just is there information present, but exactly when and over how much time is the neural response most informative about each parameter. And then we could essentially compare the optimized decoding parameters for example, encoding the timbre and compare them to those that maybe encoded the pitch. So the first thing we observed was that neural units were sensitive to vowel identity across multiple dimensions. So in these kind of stacked histograms here, you can see the green units represent the proportion of units that actually provided information about a vowel invariantly across other stimulus dimensions. Although again, I should say, if you're sort of trying to mentally compare this to the anesthetized data set, because of the optimization in this decoder, the decoder will search out the time period of the neural response where invariant information is present if it's available. Other neurons had other neurons had information about the orthogonal variable, so either about pitch or location or level, and a smaller proportion of neurons had information about both of these features. And if we zoom in on this population of neurons, and we ask across the population of neurons we had, where was the information distributed in time for the vowel information and the pitch information, or the location in the vowel, or the vowel and the level, we can see that for these three parameters that the animal could generalize across, there was a nice, clear differentiation in time about where this information was available. Actively discriminating timbre further kind of emphasized this, this timbre information being early kind of idea. So if we compare the timing of vowel information in the same neurons, same animals, when the animal's either passively listening in green or engaged in yellow, you can see that the timing of the vowel information is essentially accelerated when the animal's actively processing the sounds. And that's not seen for the pitch information in the sounds. This was unchanged. So I said the fact that the animals couldn't discriminate the whispered sounds was quite helpful. So these failures in perceptual constancy mean that we can look for neural correlates that also, or we can look at what we believe to be neural correlates of the perceptual constancy. And if they really are, they should fail too. And kind of in keeping with that, whispered sounds elicited the smallest proportion of vowel sensitive units. Those units that were sensitive conveyed less information. And if we looked across the timing of the information about the vowel in units recorded in each of the 4 different tasks, the timbre information is really clearly delayed in the units that are encoding information in the whispered case. And we have good reason to believe that really the animal only pays attention to this first vowel anyway. So the fact that there were neural units available that had information about the spectral envelope later in the response kind of fits with the fact that the animal doesn't really do the task, because early on there's very, very little information about spectral envelope available. So the third kind of short summary before I tell you one little last bit about how spectral envelope might be maintained in noise is that we can train animals in a task, but they struggle to generalize what they've learned about harmonic sounds to these whispered or noisy sounds. And these whispered sounds also disrupt the encoding of timbre and auditory cortex. But a proportion of units throughout the auditory cortex do convey timbre information in a way that is consistent with perceptual constancy, whereas others are capable of conveying timbre information, but you have to know which time point you should consider if you want to extract that unambiguously. Okay, so everything I've talked about so far has been in the sort of artificial lab-based scenario that we often find ourselves. The animal's in a quiet sound booth, we're presenting single sound bursts and asking them to make a sort of one-dimensional discrimination based on them. Of course, when we're listening, we find ourselves in much more complex environments, And we know that a key challenge for the auditory brain is to take the kind of sound mixtures that we find in the real world, to segregate them into their component sources such that you can choose which of these sources you might care to listen to. So a question we had was, how is this sensitivity to timbre maintained in noise? So again, we took the same task. This time we embedded these two vowels in a short burst of temporally restricted noise that began when the first vowel turned on and finished when the second vowel turned off. This impaired the animal's performance. So you can see that they're above chance, but there's a significant drop here. For the sake of time, I'm not showing you the whole signal to noise ratio functions, but we tested at a variety of signal to noise ratios, so we could assess thresholds as well as percentage correct measures. We then sought to determine to what extent you really needed auditory cortex to perform these judgments. I sort of motivated this talk by saying, well, isn't auditory cortex an interesting place to look? But that doesn't mean it's actually doing the job. So we trained 2 animals in this task, and we implanted cooling loops over auditory cortex that meant that by cooling down auditory cortex, we could reversibly switch it off. Somewhat to our surprise, it turns out that to perform this vowel discrimination, you don't need the region of auditory cortex that we silenced. So cooling auditory cortex left animals unimpaired in this vowel discrimination task. However, it really substantially impaired their ability to make this discrimination in noise. Now, some modeling that we've done to sort of mimic the effects of cooling, where either we took neural units and sort of subtracted or removed spikes, eliminated spikes from a response, or eliminated a percentage of units from the population, illustrate to us that the population decoding measures that we have to identify the spectral envelope of a vowel are much more vulnerable to this sort of inactivation in the presence of background noise. So you can see that in the clean conditions, our population decoder can actually do quite well with a reasonable proportion of neurons inactivated. So we believe that actually we may see a deficit in this case if we were able to really silence the whole of primary auditory cortex, or even the whole of auditory cortex. Whereas in noise, the population representation itself is much more fragile, so disrupting even a small proportion of the neurons is problematic. So how does noise disrupt decoding? Well, there's sort of two explanations that we have observed. So the first is simply that if you have a neural unit, so this is a unit that has a low frequency tuning, in normal conditions, in clean conditions, it responds nicely to the vowel, and this response, if we subdivided it by vowel type, would be quite informative. When we add the background noise, you can see the spike rate is elevated throughout the trial, and we disrupt the encoding of the vowel. So this additional noise prevents the unit from being sensitive to the spectral envelope. There's a second category of unit that we observe, and these are units that, if you can make out the black line here, which is the clean case, actually don't respond to our vowels at all ordinarily, but when we turn on the noise burst, they respond strongly to the noise burst, and these tend to be high frequency units. So we think it's really a two sort of faceted problem. One, you disrupt the information that should be available in the subset of units that normally tell you about spectral envelope. But secondly, any sort of population decoder that's looking down on some kind of frequency representation across auditory cortex is going to be strongly disrupted by a population of high frequency neurons that suddenly start firing in this case. So we know that noise recruits these additional units and triggers extra spiking in vowel sensitive units. So this is the firing rate of all neurons across time. But we can see that this effect, so the fact that in noise, the red PSTH is higher than the black, it's pretty much gone by the second vowel sound. So it seems like there may be adaptive mechanisms there in place. And we can see this too in the population decoding. So you can see that in the clean case, decoding performance is high in response to the first vowel, much lower in the noisy case, and no longer statistically different in the case of the second vowel. So it seems like there's some rapid adaptation that might be restoring timbre sensitivity in these, this was single unit responses, an average of a population of them, and this is the population decoding. They essentially show the same thing. And this is the animal's behavior. So again, you can see that the behavior matches the decoding performance in the first vowel and not in the second vowel. So we also asked, could we actually recover both behavioral and neural performance through adaptation by, instead of embedding the vowel in a restricted noise, by having a continuous noise background? And now if we look at the thresholds, so here, the lower the threshold, the better you are. All of our animals have better thresholds in the continuous case than the restricted case. Our single neuron performance recovers. And this additional spiking, which we see in the red case, which is the restricted, is now lost. So in the continuous case, we actually see a sort of suppression of the cortical responses. And the population coding too is recovered. And actually this particular feature is not dependent on auditory cortex. So when we silenced auditory cortex, we didn't see any effect in performance in the continuous noise case. So to summarize what I've told you today, auditory cortex is required for performing spectral timbre discrimination in the presence of a temporally restricted noise. So whether that's because the role of auditory cortex is actually to pull apart those two sound sources, or whether it's because it's simply that the representation of the spectral envelope is more vulnerable in the presence of noise, and that means that an auditory cortical impairment is more substantial, is kind of yet to be determined. We observe these really rapid adaptive mechanisms that mean if you have a background noise, then really within a couple of 100 milliseconds, you can adapt to that noise, and your auditory cortical neurons can restore their ability to extract spectral timbre. And really, everything that we learned in the awake behaving animals, we'd already seen in the anesthetized animals. So this is a case where kind of doing things in the proper, correct way didn't actually buy us that much more information. Information is multiplexed. If you look at the right point in time, you can extract invariant representations. And my final kind of conclusion is really, I think animal models can be really useful for providing insights into general mechanisms of timbre perception. All of this work, so the noise study is currently on bioRxiv in review with Stephen Town as the first author, and he's really spearheaded all of this work in my lab. And the first bits of the work I was talking about was with Kerry Walker and Andy and Jan. Thank you to the people who funded us, and thank you for you guys for listening.
Thank you, Jenny. That was a great talk. Just to remind people, yeah, if you want to ask a question, please raise your hand on the, yeah, or with the camera, or also in the chat, so that I can get an overview of who wants to ask. So yeah, maybe we can start actually with Peter.
Hi, Jenny.
Hi.
You know, I'm an admirer of your work. I think you're doing exactly the right experiments for getting at the neural coding at the auditory cortical level. And basically, I think your findings blow away all of the prevailing ideas that about rate coding of individual features and point to some sort of multiplexed population code. And the third thing, if I understand your results correctly, suggests a spike latency code where the timing of spikes and the particular neurons that are responding at particular times is a critical thing.
Yeah, I think that's fair. Yeah, I think, I mean, in the auditory system where time is just so critical in the stimulus domain, it seems kind of insane that we would really take a kind of a more visual cortex approach, if you like, and just consider, you know, long spike rates. So I think, yeah, the more we dig down into the timing, the more we kind of learn about how, I guess, how all of this is potentially fed into a population code, which is capable of extracting these things at different times.
You know, it might be that if they looked at the visual cortex the way you're looking at the auditory cortex, a lot of problems that they have might be solved.
That's also true, but I wouldn't dare tell them that.
Okay, thank you, Peter. I think we have a question from Kai.
Hi, Jenny. Thanks for this great talk. I just have a small comprehension question regarding the last bit of your talk. So you were presenting these vowels and noise, and I was wondering what were the intra-onset times of the noise and the vowels in the concurrent case, so in the non-adaptation case?
They were exactly concurrent with the, but because we have these two identical vowels, because when I originally designed this experiment a decade ago, I wasn't convinced they'd be able to learn the task. So I thought I would make the task easy for them and give them kind of 2 bites at the cherry, which turns out was probably not a sensible design choice and not a necessary design choice. But so the restricted noise came on precisely at the same time as the first token of the vowel and persisted until the offset of the second token of the vowel.
Mm. So you're, in that sense, you're conflating the onset, some of the onset cues, right? So this might be, then it seems tricky to then just argue for the role of adaptation with the other condition of continuous noise, right? Because
yeah, I mean they're sort of confounded in multiple ways, right? Because adaptation may be one of the ways in which you do separate different sound sources, or it may be one of the ways in which you pull signals out of kind of stationary noises. And we have some follow-up data that we recorded and have yet to make sense of, where we were more systematic in varying the onset of the noise and the vowel. But the problem is, it's really, yeah, the grouping cue is so strongly tied up with the onsets. And as soon as you provide any time before the vowel in order to separate out that onset cue, then you provide for adaptation. So it's a tricky experiment to work out how to how to really unambiguously kind of argue in one direction or the other.
Okay, thanks.
Thank you. Laurel Carney has a question.
Yes. Hi, Jenny. That was a great talk. You summarized a lot of information very quickly. I have a question about the generalization aspects for both the behavior and perhaps the physiology. You mentioned that the animals were kind of quickly retrained in a new task, but if they're really generalizing, they should be able to do it either with no retraining or when the stimuli are intermixed. Did you ever try that, or were the stimuli always blocked?
The stimuli were blocked, but we have enough animals now that we can look at the learning curves, in inverted commas, from the first trial that they experienced a different sound or a different, and there is no learning curve, except so location screwed them up a little bit in that sometimes they needed kind of 6 or 7 trials. Mm-hmm. But even 6 or 7 is not many in the scheme of things. It's just that I think as predators, they just have such a kind of innate response to go towards the location of a sound source that they couldn't quite help themselves the first couple of times. But for all of the pitch stuff, we never really had to train them as such. They more or less did it on the first trial. It's hard to prove in a single animal, but across a cohort of animals, we can see it's pretty convincing.
Do you think they could do it if they were really randomized, sort of interleaved?
So, well, yeah, so once we start changing, we train them with a fixed F0, mm-hmm. and then once they've reached criterion, we randomize the F0 across the whole range. So yeah, it's not like we train them on 200 and then we add in 250 and then we add in 300, and they cope fine with that. So I do think they're generalizing reasonably naturally.
Okay, thanks.
There is a question in the chat by Daniela Lenti. Is there any evidence of individual acoustic recognition for ferrets in the wild?
As in recognition of individuals? I guess that must, there's very little data actually about the acoustic calls of ferrets in the wild, or indeed, there's a little bit there's one study on Siberian polecats, which is basically like some guy caught some in the wild and then poked them with sticks and observed that they screamed. But in terms of like natural communication within a group of animals, there's really disappointingly little information based on that. I mean, much as I like to argue that ferrets are vocal animals, I don't think they're very sophisticated vocal animals. So I think they probably rely more on smell to identify individuals, but that's really just me speculating based on observing them rather than any kind of evidence.
I see. Thank you. And then there is a question by David Dalbom.
Hi, thank you. I just wanted to ask, and this is particularly concerning the first study where you were showing the various levels of mutual information and sort of degrees of localization for pitch and timbre. And it looked like timbre was more heavily localized and certainly had much higher mutual information numbers than or timbre did than pitch. And do you have any ideas, speculative or otherwise, what that says about perhaps coding strategies or, and I suppose that's just it. Thank you. Yeah,
so your observation is bang on. Like the amount of timbre information was quantitatively higher than about pitch. Kerry Walker, in conjunction with Josh McDermott and Andy King, has done a really nice study trying to get to the bottom of what cues the ferrets use to do pitch discriminations. So the ferret kind of cochlear bandwidths are wider than humans. They're more sort of guinea pig-like even than cat-like. So they have a relatively kind of smeary frequency representation, which means that they seem to rely more on temporal cues to extract pitch than spectral cues. And my suspicion is that's probably what underlies it. They certainly have access to pitch. If, for example, if I present, instead of a target vowel with a background noise, a target vowel with a competing vowel, if I give them a pitch difference between those vowels, they behave like humans and segregate the two vowels and identify the target and respond to that. So they can use pitch, but I think they do so in a slightly different way from how humans do. And that probably results in there being less information available to them, or less easy information available, perhaps.
Do you see it as all possible that pitch is perhaps more widely distributed, or the code or is it just, do you think it's just that they don't have very good representation of it?
No, I mean, I think they have a reasonable representation, and in some ways I think those mutual information plots are a bit misleading because of the different scales. And there's one particular field that really shouts at you for the timbre information, which makes it look quite localized, but actually there's a lot of information distributed across all of the areas. Mm-hmm. Pitch information looks more distributed, perhaps because I think it's clustered as it would in a marmoset, kind of on the low frequency edges of the primary and non-primary areas. So if you visualize it averaged across a field, it looks very distributed, but it is tied to those low frequency areas. And by definition, because of the organization of the ferret cortex, knowing exactly which field you're in is very tricky, and that melting pot in the middle. And that's where most of the pitch is. So my suspicion is that actually pitch is probably more localized, but it doesn't come across like that in the way that we were depicting the data earlier in the talk.
Gotcha. Thank you very much.
Kai would like to add something on that.
Just had another question because it's so interesting. I was fascinated by this time course you showed, the time course of timbre representation towards the beginning of the talk. I was wondering whether you could speculate perhaps a little more about why this might be the case, that the timbre is so early on represented.
So I mean, there's a trivial explanation which would account for like 10 milliseconds of the 70 or something, which is that to extract the pitch, especially if you're going to do it over using temporal means, you need at least 2 cycles of the waveform. Whereas for timbre, if you've got a snapshot of the whole tonotopic array, you could extract it instantly. I mean, even if you added a delay for every kind of processing station, it's not really, the delay in the information still seems to be greater than that. So I don't think that's all of it. And I don't have a good explanation for it.
I mean, there are analogies, of course, in human behavior, right? There are these studies from Daniel Presnitz's lab showing that you can categorize timbre and within like 8 milliseconds of stimulus length and so on. And also acoustic information in natural sound sources seems to be really rich at the beginning of sounds. But it's interesting to see that in these neural data.
Yeah, I agree. I still don't have good explanations, but.
Yeah, actually, I also wanted to ask something related to that particular work. So, are you aware of any similar research in humans that you could compare with this data you have on the ferrets?
Yeah, so there's a series now of papers from Emily Allen, I think, in Andy Oxenham's group, where they've been looking at sort of joint pitch and timbre encoding, and there are a lot of, I can't remember, I mean, she's moved more towards, I think, orchestral sounds now, but there are some really interesting parallels there, and there's nothing yet that they've published that really stands in kind of stark disagreement with what we've seen in the kind of single neuron data. Jan, in collaboration with Micah Murray's group in Switzerland, did a kind of EEG study using almost exactly the same stimuli, but asking people to changing the vowel identity and the pitch and the location from trial to trial, but within a block, asking people to pay attention to changes in only one of those dimensions. And they showed some kind of interesting differences in terms of the representation of information if you were doing an identification task rather than a localization task, sort of feeding into kind of what and where theories. So yeah, there is some stuff out there that's quite closely related.
Okay, thank you. So are there any questions? We could use one more question if anybody has one. I see none. All right, so I think we can conclude Jenny's keynote. Again, thank you very much. It was great to have you here.